All right, these are your directions for performing your BerryCoolDNA lab tomorrow. Okay, the purpose of this lab is to observe DNA from real living organisms, and we are using strawberries. We have done this lab with other cells. We've used peas, you can use bananas, but we like to use strawberries because they are chock full of DNA. They have 7 chromosomes, but they have 8 copies of those 7 chromosomes. So total, there's 56 chromosomes. So that gives us a lot of DNA to work with. So this is what your station will look like, although you guys will be sharing microscopes in between stations because Ms. Strawn and I are doing the lab the same day, so we just have the one set of microscopes. And the only thing you really won't find at your lab, well, 2 things. The strawberries will be located in a central location in the room. You just need those right at the beginning. And then you'll also need some rubbing alcohol that has to stay cold. So you'll have to get that from a central location in the room as well. But you'll have a plastic bag. You'll have some extraction buffer, which is in the beaker here. Just circle that for you. The extraction buffer is basically a mixture of shampoo and salt and water. You have a graduated cylinder for measuring it. I mentioned the rubbing alcohol will be in a centralized location. Paper towel, rubber band, test tube and rack, wood splint or a dropper, and then microscopes and slides, because ultimately you guys are going to be pulling DNA out of the cell and then be looking at it with the microscopes. So here's the basic procedure. You're going to put the strawberry in the bag, in the plastic bag, and zip it up. And then you're going to mash up the strawberry for 2 to 3 minutes, okay? At that point, you're going to open up the bag and you're going to measure 10 milliliters of the extraction buffer. That's the shampoo solution. And you're going to pour that into the bag. And you're going to mix that together for another 1 or 2 minutes. And try not to make too many soap bubbles. Just make it not too foamy. You can mash it. You don't need to pound on the bag or shake it too vigorously. Just mix it gently. Then you're going to set up a little filtering apparatus, and we'll show you what this looks like before you start the lab tomorrow. But basically, your paper towel is going to go on top of your plastic cup secured with a rubber band. And then you're going to pour the juice from your bag through that paper towel. And the goal here is to keep all of the cell parts and the pulpy part of the strawberry and the seeds in the paper towel, and just the juice comes through the paper towel. This part takes a little bit of time, so please be patient. And, you know, you can answer some of your questions on your lab while you're waiting. Whatever juice you end up with in your cup, and hopefully you have a nice centimeter from the bottom, you're going to pour that into your test tube. And then at that point, your paper towel can go in the garbage, and so can your baggie. And then you're going to be adding some rubbing alcohol to your test tube. So you want to pour it really slowly. So you're going to tilt the test tube to the side and pour in the rubbing alcohol slowly. So it's about halfway full with liquid in total. And you should see that there's 2 distinct layers. You'll have a rubbing alcohol layer and a strawberry layer. And you're going to let that sit for about 2 to 3 minutes without disturbing it. And again, you can answer some of the questions on your lab while you're waiting. And you're just watching for the formation of this kind of whitish, mucusy stuff. Sometimes people have compared it to snot. That's kind of what it looks like, or egg white. And that is the DNA. And you'll notice that it starts to float up to the top. So at that point, your goal is to take that substance out of your test tube and put that on a slide. So we can use a wood splint for that, or a dropper. The caution with the dropper is that it tends to get stuck inside there. So you want to do that as carefully as you can. You might need to use 2 wood splints, one to wrap it around and one to kind of scrape it off onto your microscope slide. Just kind of think about wrapping it around and around and around here, kind of like spooling cotton candy. So try to keep it at eye level and watch what you're doing. Do it very carefully. So here you can kind of see what that looks like. Again, it looks kind of like snot or egg white. So then you are going to put that blob on the slide, and you're going to sort of tease or stretch that apart using the toothpicks or wood splints, okay? You're going to put a cover slip on it, and then you're going to look at that on the microscope. When we put on the microscope, you want the light setting really low because this doesn't have a lot of color on it. If you pass too much light through it, it'll be difficult to see. So you're going to put a cover slip on, look at it. The rest of your materials can get rinsed out. Your dropper, the plastic cup, the test tube, plastic bag can go in the garbage. And when you're all done with your slide and cover slip, those can get rinsed and left off to dry as well. All right, when you're done observing and you've looked at the DNA, you are going to clean up. So paper towels and baggies go in the garbage. Cups are rinsed and left upside down. Test tubes are rinsed, left upside down in the test tube rack. Slides and cover slips are rinsed, and maybe leave those on a paper towel for the next group. And then you're going to answer your concluding questions. Use your notes if you need to help you answer the questions, and then you'll turn in your paper. And then there's other work you could be doing if you are finished early. One more little thing that I'll just mention is remember that our microscopes only go up to 400x. So you're probably not going to see, the actual double helix because our microscopes are not powerful enough to see things on a molecular level. But think about one of the questions on your paper is what are you actually seeing and what does that look like? So that's something we'll discuss after we've done the lab. Thanks very much, and let me know if you have questions.